one over right now. Okay, so today in Screencast 4 or 5, I've lost count, we're going to take a look at the types of RNA, but mainly we're going to be taking a look at the first portion of the synthesis of a protein called transcription. All right, but first the types of RNA as a review so you can have it organized in your notebook. What does RNA do? As a very generalized definition, it functions to transfer genetic instruction from the nucleus to the cytoplasm. The hard part about it is that DNA is a molecule that is too large to leave the nucleus. So the instruction that it needs to make protein has to leave, and so it does through RNA specifically, ladies and gentlemen, through the type of RNA called messenger RNA. Messenger RNA holds the code. It is often called the codon. Why? Because the code's on it. What kind of code? The code to make a protein. More on that coming up in just a few minutes. All right, let's get the next kind on there. All right, so you've got transfer RNA. Transfer RNA carries the amino acids for ribosome. So transfer RNA, super important protein synthesis coming up. Transfer amino acids to make a polypeptide. And a ribosomal RNA or rRNA is simply the molecular material that make up a ribosome. So ribosomal RNA. All right, all to satisfy the central dogma of biomolecular biology, and that is the synthesis of protein. DNA to RNA to protein. All right, so we're here to discuss really the synthesis of protein today. And the synthesis of protein contains two parts. Number one, transcription and translation make up a protein synthesis. So DNA controls the production of a protein. Let's give you a super overview of it. Then we'll take a look at this diagram on a larger panel. It's a good one. I want this to go into your Cornells. All right, so transcription is a process that takes place entirely within the nucleus of a eukaryotic cell. And what it does, it transfers the genetic information or the code from the DNA to the RNA, specifically to messenger RNA. So transcription, DNA, the code onto messenger RNA through the same kind of process that happens if you are copying DNA in the S phase. Although we're not going to get really deep into this, the exact process that it is covered in your textbook, but we're not getting in deep to it. We're doing 9th grade version of it. DNA is transcribed. Important to know that transcription happens in the nucleus of an organism. All right, translation, the other process which takes place at the ribosome to basically translate the code into the protein. So you transcribe, then you translate. Transcription, translation, protein synthesis. In fact, an IB fun fact is that they don't even use the word protein synthesis, two separate processes. All right, let's take a look on your next panel. Again, you have a eukaryotic cell where you see transcription taking place, DNA to messenger RNA. The messenger RNA is small enough to snake its way through the nuclear pores, doesn't go through the phospholipid bilayer. That's the main function of why a nucleus has holes and it has pores in it, so that messenger RNA can squeeze its way through. Messenger RNA links up at a ribosome with transfer RNA for the process of translation. So that is what we're going to be taking a look at today. So continuance of a species. And this overall is called expression of a gene or gene expression. So let's take a look at transcription right now. All right, remember, DNA is the code. DNA cannot leave the nucleus. Unable to. The molecule is too large, double helix. So it transcribes onto RNA, which then is translated to make a protein, and then your gene is expressed, okay? Gene, bring it down here. Gene expression. Gene expression right there. Right here. Gene expression. DNA to mRNA to protein to a trait. All right, sequence of amino acids, shape and function. This goes back to November with us. Remember, amino acids then shape into a protein. Proteins have function. All right, so DNA is the code. You got to keep it in mind. DNA is the code. All right, again, the RNA forms and things along those lines in chapter 25. So gene expression, transcription, translation, making of a protein. So during transcription, all right, simply put, we're going for big picture here. All right, you synthesize a copy or a strand of messenger RNA copied from DNA, okay, by an enzyme called RNA polymerase. All right, RNA polymerase. So during transcription, you're going to take a look here. You're going to listen here. All right, you're going to take some hardcore notes from the voice notes here. All right, a segment of DNA that we call a gene, okay, serves as a template. Remember, a chromosome has a lot of genes on it. All right, so if you take a look at this one right here, we need to actually learn how to read this molecule here. All right, so let's get rid of my linings here. Okay, get rid of, should have had that erased before you came in. All right, so you'll notice the blue template, which is your strand of DNA, your sequence that needs to be read. All right, so during that process of producing the messenger RNA, it is read, all right, and produces a complementary strand of messenger RNA. But remember, the complementary strand is different because the U takes the place of the T, and these two strands never actually bond together. All right. So previously, molecular genetics really thought that actually protein was the G, but now we know. So all three types of RNA are used. So what happens? RNA polymerase separates the DNA strand and synthesizes a complementary strand of RNA from one of the DNA strands. Remember, this is entirely other. That's why you need the 3 to 5. Look at that there. That's why we covered that in earlier screencast. So you have a 3 to 5 directional RNA, DNA. Your RNA is going to be 5 to 3. Forget about the translation portion at this time. All right, so each 3 nucleotide bases code for an amino acid. Okay, each three, I want to keep that in mind. So you see UGG, UUU, GGC, UCA. This doesn't really mean any protein at this point. This is just an example. So RNA polymerase separates the strands and synthesizes. When the DNA strands are separated, you have ribonucleotides, which join. All right, so the UGs, ribo, the pink colored there. All right, once the strand is synthesized, the polymerase attaches from the DNA molecule, the double helix then reforms. We're really into this. So the code is on the DNA. The DNA is transcribed. So holding the code on the messenger RNA, what is it going to be the code for? Amino acids, which are going to make protein. You got that? Transcription, DNA to messenger RNA. That is transcription. You can see it again in this picture here. The RNA polymerase here is the big bubble around it. All right? You see the DNA. How do you identify the DNA? Anytime you see a thymine, it must be the DNA. DNA then, again, you see the transcription of this strand down here. Just transcribing, adding RNA nucleotides to it, the U taking place of the T, the process of transcription. So here, what you would do then, you might be prescribed in a multiple choice question with a 5 to 3 strand of DNA and asked to transcribe that DNA. So how would you do that? How would you do that? Simply, if it's a multiple choice question, find the corresponding, at least for this one, 3 to 5 line. And that's the corresponding complementary line. All right. And again, remember AT in DNA, AU in RNA. All right. Continue Continue chewing this down. There's no difference with C and G. So you complete this strand for a second before I do. All right, can I give you a second? Quiet myself down. Okay, I'm back. That means you stop while you figure out the rest of this. All right, U, A, all right, C, G, C, A. My handwriting's horrible. Stick with me. Engage, engage. Can't even stay on the line. Gosh. And A. There is a strand of messenger RNA. So you have your, back to the pen where you can, right? So this is the half a strand of DNA right here. And you have the messenger RNA. Make that smaller right there. All right. So let's continue the learning. I think you can do that. You need to be able to do this. You need to be able to do this. So let's continue the learning here. All right. So one thing that you should know again, the sequence of the DNA transcribes is gene. And now we're going to, you see below the word codon. Didn't even know what I made sense there either. All right. Separate each three. All right. Now notice you're going to have a little left over on the end. We often do that, or else I could have left one out. Maybe I'll add another line here. No, I won't. All right. So what is a codon? A codon is found on messenger RNA only on messenger RNA. All right. So we're trying to learn how to translate the genetic code coming up. All right. So codons are 3 bases of messenger RNA. So a UAG is a codon. A UAC is a codon. GCA, UAC, all right, not the UA. You need another letter there. So we're going to X this out right there. All right, 3 translate into. So for instance, as you see on the line there, AUG, CCC. Again, you have 4 letters, UACG, however many combination of letters that there can be, and we'll see. All right, so one codon will then correspond to one amino acid. But how do you tell which amino acid it codes for? Well, we have a fairly easy way in biology to do that, all right? And that is by reading the genetic code, which is the set of rules by which information encoded on the messenger RNA is converted into amino acids and then into the polypeptide. All right, so the genetic code corresponds to